Hello, my name is Jewel Mitchell, and this is my exam mini video number 5 for Genetics 311. So, what are mutations? A gene mutation is a mutation that affects a single gene or locus. We categorize mutations on the basis of their molecular nature. However, we also encounter some terms that relate the causes and phenotypic effects of mutations. So the simplest type of gene mutation is a base substitution, which is the change of a single nucleotide in DNA. There are two types of base substitutions, transitions and transversions. In this figure are the possible changes for each. Here's the original coding strand that we're going to start out with. Since the original strand was in a 3 to 5 direction, we have to flip it to make it into a 5 to 3 direction from left to right. After undergoing transcription, all of the T's would then turn to U's. So thymine would turn into uracil. So we start out with the start codon, which is AUG, which codes for methionine, and we keep going until we find the stop codon. So the next one would be GUC, which codes for valine, AGC, which codes for serine, and AAG, which codes for lysine. These are the 4 amino acids that the protein forms from this DNA sequence. We don't count the stop codon, which is UAA. So these are the 4 consequences of point mutations. Missense is when a base substitution results in a different amino acid in the protein. A nonsense mutation changes a sense codon into a stop codon. A silent mutation is a change in the nucleotide sequence of DNA that does not alter the amino acid sequence of the protein. Readthrough is when a stop codon is changed to a codon that codes for an amino acid, resulting in a longer protein. So here's an illustration of each single nucleic acid point mutation. We have DNA strand and then we have an mRNA strand. So GUC to GUU would be a transition, AGC to AGA would be a transversion, AAG to UAG would be a transversion, and UAA to UUA would be a transversion. Phenotypic effects occur from these mutations where different tRNAs bind to different synonymous codons. Which synonymous codon is used can affect the rate of protein synthesis.